First, I would like to thank the organizer for this great opportunity to present in this meeting. This has been a collaborative project where we identify a new variant in the gene encoding for alpha-2 delta-1, the calcium channel auxiliary subunit. Calcium channels have been divided into CAV channels have been divided into three groups. Only CAV1 and 2 are known to be associated with the beta and alpha-2 delta subunits. So this is a schematic of the channel complex. This is the pore-forming subunit, also called the main subunit. It is associated with the cytoplasmic beta subunit shown in here. The beta subunits regulate the surface expression and the gating properties of the CAV channels. And the alpha-2 delta proteins shown in here, there are 4 alpha-2 delta. There are a GPR anchor protein. They have a very large extracellular domain which interact directly with the loop between S1, S2 of the main subunits. Alpha-2 delta-1 is important. It increased the density of CAV channels at the plasma membrane. It also targets the channels to specific subcellular sites, such as neuronal processes, and also it increased the function. Interestingly, there are a number of new research showing that in addition to this role as auxiliary subunit, alpha-2 delta-1 has roles independent of calcium channels. So it's not surprising that effect in expression of the alpha-2 delta protein have been involved in pathologies. And these are a few examples for alpha-2 delta-1 and alpha-2 delta-2. So firstly, alpha-2 delta-1 has been shown to be this time upregulated in DRG neurons in neuropathic pain models. And also another interesting thing is gabapentinoids, which are anti-epileptic drugs used in neuropathic pain, binds directly to alpha-2 delta-1 and alpha-2 delta-2. In the other hand, loss of function of alpha-2 delta-1 have been involved in 2 different pathologies associated with epilepsy and intellectual disability, and mutation associated with cardiac pathology. For alpha-2 delta-2, also loss of function mutation have been involved in epileptic encephalopathy and cerebral ataxia. Alpha-2 delta protein undergo multiple post-translational modification, which make them unique proteins. I'm going to talk mostly about alpha-2 delta-1, but most of the features are shared among all alpha-2 delta proteins. The EM structure published in 2016 showed a great detail into the organization of the domains in alpha-2 delta-1, where the VWA domain interact directly with the alpha, the main subunits of the calcium channel complex, and there are the 4 cache domains shown in here, which function is still not known. Another interesting feature of alpha-2 delta-1 and all alpha-2 delta, these proteins are cleaved. So they are encoded by a single gene, but they are cleaved into alpha-2 and delta peptides that are linked by disulfide bonds. So this is a schematic of the alpha-2 delta-1 that is uncleaved. Disulfide bonds are formed between the alpha-2 and delta before cleavage. So after cleavage, these 2 moieties stay linked. In a Western blot, overexpression of alpha-2 delta-1, this is how it looks, because alpha-2 delta-1 is also heavily glycosylated, so we cannot distinguish between the cleaved and uncleaved. So for that, a treatment with PNGSF, which remove the glycan, allow us to distinguish between the alpha-2 delta-1, which is the uncleaved form shown in here, and the alpha-2 form here, the cleaved one, as the antibody recognizes the alpha-2 peptide. So in this study, there have been 2 patients that have been identified and show a similar phenotype with an early-onset epileptic encephalopathy, neurodevelopmental disorder, and interestingly, insensitivity to pain. So a sequence in alpha to delta 1 found in, first I will focus in patient 1, that the patient 1 is homozygous for a frame shift mutation. So both parents carry the same mutation, but the parents are healthy. On the other hand, the patient 2 is heterozygous for a truncated frame shift that occur quite early in the alpha to delta 1 gene, and a missense mutation, which is a glycine in the position 209 to aspartate. And same thing, the parents are healthy. So it's interesting that, so this is a collaborative project. So because of the similar phenotype between patient 1 and patient 2, we were interested in knowing whether the alpha to delta 1 is expressed. This is the relative mRNA of fibroblasts from 3 healthy patients compared to the patient 1 and 2. This has been done in the exon 6, 7, and exon 10 and 11. Just to remind you that the patient 1 is the homozygous for a frame shift variant, and you can see that there is little RNA messenger for alpha to delta 1, indicated that there is a missense, the degradation of the RNA messenger for alpha to delta 1. However, the patient 2, which is the heterozygous frame shift and missense variant, show a good amount of RNA messenger for alpha to delta 1. In the protein level, the same thing, fibroblasts form healthy, 3 healthy controls, and patient 1 and patient 2 were used. And this is the example of alpha to delta 1 and tubulin Western blot, and this is the quantification. And you can see that patient 2, although show a reduction in the protein level compared to the control, it showed expression of alpha to delta 1. As I said, the patient 2 is heterozygous. It's carrying a frame shift, which is quite early in the protein, so we think that is either not expressed or non-functional. So we focus our work on understanding the effect of the missense mutation, the glycine 209 to aspartate. For that, we did an alignment, and as you can see that this glycine in the position 209 is conserved in all species shown in here in alpha to delta 1. And just also to mention that it's located in the cache 1 domain, and so also is shown in here. So the first thing we did is analyzing the effect of this mutation on the expression of alpha to delta 1 at the cell surface, and this is the cell surface biotinylation showing a wild type and mutant alpha to delta 1 and a negative control. These are overexpression system. You can see that we can hardly detect the mutant at the cell surface compared to the wild type, and this is quantification of cell surface alpha to delta 1 over total shown in the whole cell lysate, and you can see there is a significant reduction of expression of the mutant at the cell surface. This was also the case by doing immunocytochemistry. The same thing, we expressed HA tagged alpha to delta 1 wild type or mutant, and using an HA antibody in non-permeabilized cells, we can detect alpha to delta 1 wild type nicely at the cell surface, and we can see that the mutant is poorly trafficked to the cell surface. It's shown here is M-cherry. We use it as transfection marker. And this is the quantification. We see, again, a significant reduction of expression of the mutant alpha to delta 1 at the cell surface. So as I said, alpha 2 delta 1 is important auxiliary subunit for CAV 2.2. So what happened for the CAV channel function? So we use patch clone technique, and this is example of current traces and IV. We express CAV 2.2 together with the beta subunit with either alpha 2 delta 1 wild type or mutant alpha 2 delta 1, and compared to the condition where no alpha 2 delta was expressed. We use this stimulation protocol, and you can see in the presence of wild type alpha 2 delta 1, we can see a nice increase in the function of CAV 2.2 comparing to the no alpha 2 delta, and this was absent in the presence of the mutant alpha 2 delta 1. And this is also clear in the IV curve. We found the same for CAV 2.1, where we did exactly the same experiment. Again, we found that the mutant alpha 2 delta 1 is not functional. It doesn't enhance. The CAV 2 currents. So the next question was whether this lack of functional expression of CAV 2.2 was due to a lack of traffic. So for that, we expressed a double-tagged CAV 2.2 with a GFP in the N-terminus, and this allows us to see the total protein that is expressed, shown in here, and the HA tag, which is in the extracellular loop, so we can detect what is at the cell surface. Again, in all conditions, beta subunit was expressed with either alpha 2 delta wild type or mutant, and compared to no alpha 2 delta condition. And as you can see, in the presence of wild type alpha 2 delta 1, we can see a nice expression of CAV 2.2 at the cell surface, comparing to no alpha 2 delta. And this was again not the case in the presence of the mutant. And this is the quantification of the HA in gray and the GFP in green, where we can see that there is no difference between the condition where the mutant is expressed compared to the no alpha 2 delta condition. Interestingly, we also saw a decrease in the GFP, which represents the total protein that is expressed. We think that it's probably related to ER degradation. So we concluded that the G290 impairs the ability of alpha 2 delta 1 to enhance CAV 2.2 trafficking to the cell surface. So what happened in neurons? These are hippocampal neurons that have been transfected exactly with the same construct, double-tagged CAV 2.2, together with the beta subunit. M-cherry was used as transfection marker. We used exactly the same condition, plus wild type alpha 2 delta 1, and mutant alpha 2 delta 1 compared to no alpha 2 delta condition. And in the presence of wild type alpha 2 delta 1, you can see a nice expression of CAV 2.2 in the neurites, and this is in the HA and GFP signal, whereas we can hardly detect CAV 2.2 in the neurite where no alpha 2 delta is present, and this was exactly the same for when the mutant is expressed. Again, this is the quantification. In gray is the HA, and then in green is the GFP. We can see that both HA and GFP are reduced when comparing in presence of the mutant alpha 2 delta 1 comparing to no alpha 2 delta condition. So again, we found that the alpha 2 delta 1 mutant does not promote CAV 2.2 trafficking into neurites in hippocampal neurons. And then we image the cell body of these hippocampal neurons. We used exactly the same construct and same condition. We can nicely detect CAV 2.2 at the cell surface in the presence of wild type alpha 2 delta 1. This was not the case with no alpha 2 delta condition, and again the same in the presence of the mutant alpha 2 delta 1. And this is again the quantification where we see significant reduction of HA signal at the cell surface in the presence of the mutant, which was not different from no alpha 2 delta condition. And also, interestingly, we could also see a reduction in the GFP, which, as I said, represent the total protein that is expressed, and it's shown in here, which also indicate an ER degradation, as we think that the protein is retained in the ER. So again, we found that the alpha-2 delta-1 G209 D does not promote CAV2.2 trafficking in hippocampal neurons. So because of this lack of effect of the mutant, we were wondering whether there is no interaction between the CAV2.2 and alpha-2 delta-1. For that, we did communal precipitation experiments where we expressed GFP CAV2.2 HA together with a wild type or mutant alpha-2 delta, and also has the HA tag. So both protein has HA tag. So by doing a Western blot, we can detect in the same Western blot using HA antibody CAV2.2 and alpha-2 delta-1. We used as negative control a CAV2.2 HA with no GFP, as we used a GFP antibody to do the immunoprecipitation. And this is shown in here, the negative control with no GFP in the N-terminus. We don't detect any unspecific band. However, when GFP CAV2.2 HA was co-expressed with the wild type, we can see a nice immunoprecipitation of the CAV2.2 together with the co-immunoprecipitation of alpha-2 delta-1 wild type, which shows an interaction. In the case of the mutant, we can detect a nice immunoprecipitation of CAV2.2. However, the co-IP was very weak. We could detect a band, but it was much weaker. And interestingly, it looks like it was higher molecular weight compared to the wild type. There was a shift in the apparent molecular weight between the wild type and the mutant. So we were wondering whether it was due to a differential cleavage. As I mentioned in the introduction, alpha-2 delta-1 is cleaved into alpha-2 and delta peptides. So what we did is we repeated the communal precipitation, and this time we treated the samples with PNGS-F, so we can distinguish between, it's shown in here, the uncleaved alpha-2 delta and the cleaved band. And this is for the wild type alpha-2 delta, and this is the mutant alpha-2 delta. And as you can see in the co-IP, we can detect both uncleaved and cleaved alpha-2 delta-1. However, we see a stronger band corresponding to mature alpha-2 delta. And this is the quantification, where in the blue, dark blue, it shows the mature cleaved alpha-2 delta, and in the light blue is the uncleaved. Here we show that there is preferential interaction of CAV2.2 with cleaved alpha-2 delta-1. Regarding the mutant, we can detect only the uncleaved alpha-2 delta-1, and we couldn't detect any mature alpha-2 delta, which indicate that there is a reduced interaction of the mutant with CAV2.2 and a limited proteolytic cleavage of the mutant. Finally, we wanted to know whether the mutant alpha-2 delta-1 would interfere with the function of other alpha-2 delta. As we know that sometimes more than one alpha-2 delta is expressed in the same cells. So for that we did patchcrumb technique, where we express CAV2.2 together with the beta subunit, the same, expressed alpha-2 delta-3. We used empty vector compared to the condition where alpha-2 delta-3 is co-expressed with the mutant alpha-2 delta-1. And as you can see that there is no difference between these two conditions, and this is shown in the IV, which shows that alpha-2 delta-1 mutant did not affect the ability of alpha-2 delta-3 to increase the CAV2.2 current, so it didn't interfere with alpha-2 delta-3 function. So this is a conclusion where we found that in patient 1, which is the homozygous frameshift variant, we think that has a nonsense mediated mRNA decay, so we couldn't, or we detect a very small amount of RNA messenger for alpha 2 delta 1. We also found that the variants in patient 1 are a combination of very early frame shift and a missense variants. We found that this missense mutation, the G209D, is deleterious mutation as it affects the traffic of the alpha 2 delta protein itself, and also doesn't increase the function or traffic of CAV2.2 channels. We also showed that the mutant show reduced interaction with CAV2.2 and also limited cleavage into alpha 2 and delta, suggesting that this mutant is probably retained in the ER as the cleavage occur after the ER, probably in the Golgi. And also we found that the mutant did not affect the ability of alpha 2 delta 3 to enhance CAV2.2 currents, and this is not surprising as we found that the interaction of the mutant with CAV2.2 is reduced. So finally, I would like to thank all people that have been involved in this project. So it was a collaborative project between different labs, and also I would like to thank Annette Dolphin and people that have been involved in this research project, and I would like to thank you for your attention.
Thank you, that was fantastic. I'm sure we've got some questions from the audience. Do you want the microphone? Someone down there. You have to come now.
Nice work, Shehrazade. I want just to ask you why you focus on CAV2.2 to see the effect of the mutation. Could have been the same result for CAV2.1, you think?
Yeah, I think maybe I went quickly, but yeah, it's here it's CAV2.2 and CAV2.1.
Oh, okay.
We show the same thing.
No, the question is on hippocampus, because CAV2.1 in hippocampus is more dominant and has more modulation than CAV2.2.
Yeah, the thing is we have the tool, like this tagged CAV2.2, which is functional, and we can detect the cell surface. For CAV2.1, we realize that if we put an HA tag, the channel is not functional, or it's not trafficked with cell surface, because it's quite tricky to work with CAV2.1. So we used, because of the tool that we had, But I guess we'll find the same because of the function.
Okay. Thank you.
Yeah.
Okay, thank you very much. I think we're up to time anyway, so we might as well leave it at that. Thank you very much.